Hello everyone, welcome back to session 5 of this chapter 2 called Sexual Reproduction in Flowering Plants. Continuing the same chapter called Sexual Reproduction in Flowering Plants, what did we study in the last session? In the last session, we studied about the pollen-pistil interaction, the advantages of pollen-pistil interaction. You have studied what is pollen-pistil interaction from the time of landing of the pollen grains on the stigmatic surface, and when the stigma becomes receptive, it will allow the pollen grains to germinate, followed by the process of formation of a pollen tube carrying the two male gametes and reaching the ovule. So that process we studied in the last session, and also we studied about the pollen-pistil, advantages of pollen-pistil interaction, where the knowledge of pollen-pistil interaction is definitely going to help the plant breeders in manipulating the pollen-pistil interaction. We also learnt about a very important technique called artificial hybridization, where it is nothing but crossing of two genetically different individuals to create a new genotype, and artificial hybridization was obtained by two very important techniques called emasculation and bagging. So as you know, emasculation was nothing but removal of the anthers, whereas bagging was covering the stigmatic surface of a flower with a polythene bag or a butter paper to avoid the contamination of the stigma with unwanted pollen grains. Later we studied about a very important process called double fertilization and triple fusion. Why was it called as double fertilization and triple fusion? Because there was one process happening called syngamy, that is the fusion of the male gamete with the egg cell to form a zygote, and the other male gamete fusing with the polar nuclei to form the primary endosperm nucleus. Hence it is called double fertilization and triple fusion because there is 3 haploid nuclei. In today's session we are going to study about a very important process called post-fertilization event, or you can call it as an event called post-fertilization event. What is post-fertilization event? The process that is going to happen after the process of fertilization. What do you mean by pre-fertilization? Prior to the process of fertilization. So what is post-fertilization? That is process that is going to happen after the process of fertilization is called post-fertilization event, and this post-fertilization event is signified by the process of formation of an endosperm and the development of an embryo. And later on we are going to study about the process of formation of a dicot embryo and a monocot embryo, right? So now let us start with this session 5 of chapter 2 called Sexual Reproduction in Flowering Plants. So what are the post-fertilization events? As I said, post-fertilization event is nothing but the event that is going to take place after the process of fertilization. What is fertilization? The process of fusion of one of the male gamete with the egg cell to form a zygote, the other male gamete fusing with the two polar nuclei to form an endosperm nuclei. So after double fertilization, the post-fertilization changes are the process of formation of endosperm. Very important, right? So one very important post-fertilization event is what? Process of formation of endosperm, followed by the development of the zygote into embryo. Development of embryo, or you can also use a word called embryogenesis. The process of development of embryo is called embryogenesis, followed by the maturation of the ovules into seeds. That is, the ovules will become the seeds and the ovary will become the fruit. So post-fertilization is the event that is going to take place after the process of fertilization, followed by a very important process called the process of formation of endosperm and the process of development of the zygote into embryo, that is embryo development, followed by the maturation of the ovules into seeds and ovary into fruit. I think all of in the last session you have studied that the zygote is always a single cell. This single cell will become a multicellular or a matured embryo like this. Right, it will become a mature embryo. So anyhow, you are going to study about this particular part in the coming slides. So what is post-fertilization event? The events that is going to take place after the process of fertilization, followed by what? The process of formation of endosperm, a very important nutritive tissue for the developing embryo, and followed by the process of development of embryo. Embryo development will not take place without the process of formation of endosperm. The embryo will make sure first there is the process of formation of endosperm, followed by the embryo development. Now, let us understand what is this endosperm. As you know, one of the male gamete fuses with the two polar nuclei to form what? The primary endosperm nucleus. So, what is this endosperm? As I said, endosperm is nothing but the reserve food material. So, this primary endosperm nucleus will undergo repeated mitosis, ultimately leading to a nutritive tissue called endosperm. So, the PEN will undergo repeated mitotic cell division to become or to form what we call it as a nutritive tissue called endosperm, and this nutritive tissue is very much important for nourishing the developing embryo. As I said, the embryo development only will take place if at all there is endosperm. So, it makes sure first there is the process of formation of endosperm. As I said, this endosperm provides nutrition to whom? To the developing embryo. So, it is very important nutritive tissue for the developing embryo. As I said, zygote can develop into an embryo only if endosperm is formed. So, endosperm is nothing but a nutritive tissue which is filled with reserve food materials. So, what is endosperm? How is it formed? The primary endosperm nucleus undergoes what? Divides repeatedly by mitosis, ultimately forming what? Forming a nutritive tissue called endosperm. And what is the function of this endosperm? It provides nutrition, nourishment to whom? To the developing embryo. So, during the process of embryo development, it requires nourishment or the nutritive tissue which is given by this endosperm. And now let us study what are the three types of endosperm based on the division of the primary endosperm nucleus. One is called the nuclear endosperm, the other one is called cellular endosperm, and the third one is called nuclear endosperm. What is this nuclear endosperm? The primary endosperm nucleus will undergo repeated nuclear divisions to form free nuclei, and that free nuclei is called free nuclear endosperm. I repeat, what is nuclear endosperm? The primary, that is PEN. What is PEN? Primary endosperm nucleus will undergo repeated nuclear divisions to form more number of nuclei, free nuclei, and that nuclear endosperm. Subsequently, the endosperm is followed by the process of formation of a cell wall. So, such cellular endosperm is, that is what we call it as cellular endosperm. First one is called nuclear endosperm, and there is a cell wall formation, such cellular endosperm. What is helobial endosperm? In helobial endosperm, the division takes place in the first two nuclei, where half of it develops an endosperm along a cellular pattern, the other half follows the nuclear pattern. One half of the endosperm follows cellular pattern, the other half follows the nuclear pattern, and such type of endosperm is called helobial endosperm. Hope you have understood what is endosperm. Endosperm is nothing but when the primary endosperm nucleus, when is it formed? When one of the male gamete fuses with the polar nuclei to form primary endosperm nucleus, and this PN divides repeatedly by mitosis, forming what? A nutritive tissue called endosperm. And what is the function of this endosperm? It provides nutrition to the developing embryo. What are the three types of endosperms you come across? One is called nuclear endosperm, second one is called cellular endosperm, and third one is called helobial endosperm. Here you can see in the picture, this is a picture of a maize seed. This is the picture of the whole maize seed, and here you can see a cross section of the maize seed. Here you can see the endosperm, and you can see the seed coat and the developing, this part is called embryo. So, this endosperm provides nutrition to the developing embryo. That is the reason what I told you all, endosperm is a very important nutritive tissue, or it is a food substance which provides nourishment to the developing embryo, right. Now, here moving on to the two types of seed which I am going to deal it in the next session. So, based on the utilization of the endosperm, we classify the seeds into endospermic and non-endospermic. What is endospermic seed? Some amount of endosperm is retained in the embryo, and it will utilize that retained endosperm during seed germination. Such a type of seed is called endospermic seed, or it is called albuminous seed. And what is the classical example for the endospermic or albuminous seed? Maize, wheat and castor. So, what is endospermic seed? The seeds which is going to retain the endosperm, which is not completely utilized during the process of embryogenesis, which is utilized during the process of seed germination. Such a seed is called endospermic seed. What do you mean by non-endospermic seed? Completely utilized during the embryo development. Before the seed germinates, it would have utilized all the endosperm. Such a seed is called non-endospermic or non-albuminous seed. That is what? These are the seeds without endosperm, right? The food is completely utilized for the development. Example, bean, gram and pea. Here you can see the structure of a bean seed and that of a corn seed, right? So, here this bean seed is an example for what? A bean seed is an example for non-endospermic, and corn seed is an example for what? Monocot. That is usually the endospermic seed. Hope you have understood this concept. Anyhow, I am going to explain in detail in the coming session about the seed. Now, moving on to a very important concept called embryo. So far we studied about endosperm. What is endosperm? Endosperm is nothing but the nutritive tissue that provides nourishment to the developing embryo. And what is it? It is one of the post-fertilization event. That is, the post-fertilization event is followed by a very important process called the process of formation of endosperm. What is the second very important thing? That is the process of formation or development of a zygote into an embryo. So here, how do we define the embryo? Embryo can be defined as a sporophytic plant present within a seed, or you can also call it as a minute rudimentary plant contained within a seed. How do we define the embryo? Embryo, you can call it as a developing individual also, right? So, this embryo can be also defined as what? A sporophytic plant present within a seed, or the minute rudimentary plant contained within a seed, or you can also call it as the structure in plants that develops from the zygote. So, as I said, zygote, after the process of fertilization, zygote is a single cell, but a diploid cell will undergo what? Will undergo what? Repeated mitotic cell division to become a multicellular embryo, right? So, the structure in plants that develops from the zygote prior to germination is the embryo. So, either you can call embryo as what? A sporophytic plant present within a seed, or a minute rudimentary plant contained within a seed, or the structure in plants that develops from the zygote prior to germination is the embryo. Now, let us study the process of development of an embryo. What do you call that? The process of development of an embryo. The early stages of embryo development is called embryogeny. As I said, the process of development of embryo can also be called as what? Embryogenesis, or you can also use the word called embryogeny. So, the early stages of embryo development are called, that is, embryogeny. And as you know, I think here you can see in this picture, this is what is the double fertilization. So, after knowing about pollen-pistil interaction, what was it? Only when the stigma is receptive. When do you call the stigma is receptive? It will recognize the pollen grain of its own type. Then it will allow the pollen grain to form a pollen tube carrying the two male gametes and reach the ovule. So, the pollen tube carrying the two male gametes reaches the micropylar end. As you know, this is the micropylar end. And we have already studied in the process of double fertilization. The micropylar end will have what? Will have the egg apparatus. Egg apparatus consists of what? Consists of 2 synergids and 1 egg cell, and in the center 2 polar nuclei, and towards the chelagel region 3 antipodal cells. So already we have studied that it is called 8 nucleate 7 cell stage. Right. So one of the male gamete fuses with the egg cell to form what? To form the zygote. Right. So one of the male gamete fuses with the egg cell to form a zygote. The other male gamete fuses with the 2 polar nuclei to form what? To form primary endosperm nucleus. Hence, the process of fertilization is happening 2 times. Hence, the name double fertilization. Why it is called triple fusion? Here it is 3N, 3 haploid nuclei is formed. Right. So, primary endosperm nucleus. And what will happen to the 2 synergids? It will degenerate or it will dissolve. Then what will happen to the 3 cells towards the antipodal cells? They will also degenerate. Right. Now, the only functional, this one is zygote and the endosperm. As I said, this endosperm is very much required for the developing zygote. Zygote will develop into an embryo only when it makes sure that first there is the process of formation of endosperm. Right. Now, here let us see, as I said, the embryo develops at the micropylar end of the embryo sac where the zygote is situated. Hope you have understood this. So, what is an embryo you have studied? Can be called as sporophytic plant present within a seed, or a minute rudimentary plant contained within a seed, or a structure that develops from the zygote prior to germination. And what do you call the early stages of embryo development? You call it as embryogeny or embryogenesis. And embryo develops at where? At the micropylar region of the ovule. Opposite to it is the chelagel region, which forms the basal part of the ovule. So, at the end of the embryo where the zygote is situated. Right. Now, let us see the process of development. Already I explained about the embryo sac, how the one of the male gamete fuses with the egg cell to form a zygote, the other fuses to form what? Primary endosperm nucleus. Now, this most of the zygotes divide only after the formation of certain amount of endosperm. The embryo, the zygote makes sure that there is a process of formation of endosperm because this is an adaptation to provide assurance for the developing embryo. So, it will be assured that yes, I am going to get the nourishing food for my development, only then it will start its development. So, it makes sure. So, the process of formation of an endosperm is an assurance for the developing embryo so that it can develop properly. Right. Now, this zygote, as I said, after the process of fusion of the one of the male gamete with the egg cell forms zygote. This is the zygote. This zygote, as I said, is a single cell which will undergo mitotic cell division to become 2 cell stage. This is called proembryo. This is called proembryo. Here this is called the basal cell. So, there are 2 cells, 1 cell divides into 2 cells. So, this is called the basal cell and this is called the apical cell. I repeat, the zygote is a single cell which will undergo mitotic division to form what? 2 cell stage. So, 1 cell is called basal cell. The other one is called apical cell. And this basal cell will undergo repeated division to around 6 to 10 cells. Here you can see this. And this is called as suspensor. This is called suspensor. Whereas the apical cell will undergo repeated mitotic cell division to become a globular embryo. So, here the basal cell will develop into a suspensor. Right. The basal cell will develop into a suspensor. Then what is apical cell? Apical cell is going to develop, undergo repeated mitotic cell division to become a globular embryo. Globular embryo will become heart-shaped embryo. You can see here, and this is how you can see after heart-shaped embryo, a mature embryo is formed. So, here you can see the process of formation of 2 cotyledons, and if it is a monocot, only 1 cotyledon is formed. I repeat. So, as I said, this zygote is a single cell. You can see here, this is a zygote which is a single diploid cell, will undergo what? Mitotic cell division, and is divided into how many cells? 2 cells. What are the 2 cells? we can call it as one as the basal cell. Second one you call it as the apical cell. And this basal cell will develop into what we call it as suspensor. How does it develop into suspensor? It will undergo repeated division to form 6 to 10 cells, continuous cells, and it is called as suspensor. The other one called the apical cell. Apical cell will get transformed into globular embryo, globular into heart-shaped embryo, heart-shaped embryo into a mature embryo. This is a very important concept from examination point of view, children. So what is it? That is, the zygote is a single diploid cell, will undergo mitotic division to form 2 cells. What are the name the 2 cells? The above one is called basal cell, the below one is called apical cell. Basal cell will undergo again division to form what? 6 to 10 cells to forming what? Suspensor. Then the apical cell will undergo again division to form what? Globular embryo. Globular embryo will become heart-shaped embryo. Heart-shaped embryo will become mature embryo. Right? So this is a very important concept to be learned. How an mature embryo is developed. And what do you call that process of development of a mature embryo? You call it as embryogenesis, or you can also use the word called embryogeny. So most of the zygotes divide only after the formation of what? Certain amount of endosperm. So this is what? This is an adaptation for what? To provide assurance for the developing embryo. The zygote gives rise to proembryo, then subsequently followed by the process of formation of globular embryo, globular into heart-shaped embryo, heart-shaped into mature embryo. This is a very important concept. So the 2 very important post-fertilization events is the process of formation of an endosperm followed by the embryo development or embryogenesis. Moving on to the structure of a dicot seed or a dicot plant, a dicot seed we can say. When do we use the word called dicot? When a seed has 2 cotyledons. When do you call a seed as monocot? Monocot we call it as when a seed has only 1 cotyledon. So here the dicot embryo consists of an embryonal axis. So it consists of what? An embryonal axis and 2 cotyledons. So what is the structure of the embryo of a dicot? The embryo of a dicot consists of an embryonal axis and 2 cotyledons. And the portion of the embryonal axis above the cotyledon is called epicotyl. So if you can see the picture here, this is called epicotyl. Here you can see the hypocotyl, you can see the cotyledons. Plumule later is going to develop into the stem and radicle is going to develop into the shoot system. Plumule into shoot system and radicle into root system. And you can see a root cap. The tip of the embryo you call it as a root cap, right? So the structure of a dicot embryo consists of what? An embryonal axis and 2 cotyledons, right? And the portion of the embryonal axis above the level of cotyledon is called epicotyl, which terminates with the plumule. As you know, it is called what? Stem tip. Root tip and stem tip. Radicle will form the root tip, whereas the plumule will form the shoot tip. So as soon as you sow a seed, one part of the seed develops into root system and one part of the seed develops into shoot system. So what is that which develops into the shoot system then? It is the plumule. And what is that which develops into the root system? That is radicle. So these two are the very important things in a developing present in a seed. So here the portion of the embryonal axis above the cotyledons is called epicotyl, which terminates with the plumule. Here you can see which terminates with the plumule, and you can see the 2 cotyledons, right? And what do you call that? The region below the hypocotyl? At the lower end, it terminates as what? It terminates as what? Radicle. As I said, radicle forms the root system and has a root cap. You can also use a word called clypetra, which protects the root tip from damage. As you know, root is a very, very important part of a plant. So, it is protected by a root cap. Right. So, this is about the structure of a dicot embryo. Hope you have understood. So, dicot embryo consists of what? An embryonal axis and two cotyledons. The portion of the embryonal axis above the level of cotyledon is called epicotyl, which terminates as what? Plumule. And what do you call the cylindrical part? The cylindrical part, that is the cylindrical portion below the level of the cotyledon is what? Hypocotyl, which terminates at its lower end as what? As radicle. Hope you have understood. This is a very important diagram from the examination point of view. As I keep on telling that diagrams in biology is very, very important. You need to practice the diagrams properly, with that to label diagrams, and particularly the label diagrams is going to fetch you very good marks. Right. Now let us study about the structure of an embryo of a monocot. The very word itself will tell you. Dicot means what? Two cotyledons. Monocot means what? Only one cotyledon. Here the cotyledon of a monocot embryo is called scutellum. Here you can see it is called scutellum. Right, it is called scutellum, and it is situated towards one end of the embryonal axis. So, what is scutellum? The cotyledon of the monocot is called scutellum, and it is situated towards one end of the embryonal axis, right. So, here you can see that it possesses only one cotyledon, and it is called what? Scutellum, and is situated where? Towards one of the embryonal axis. And here the embryonal axis has what? Has the radicle and the plumule. And here you can see the radicle has what? The root cap, and is called coleorhiza. The root cap in monocot is called coleorhiza, and the stem tip is called as coleoptile. You can see here it is called coleoptile, right. So, the embryonal axis has the radicle and the plumule, and the radicle has a root cap. What do you call it as? Coleorhiza. Then there is epicotyl above the level of scutellum. Here you can call this is epicotyl. This is called epicotyl, right. So, as I said, it is called what? Coleoptile. Coleoptile means what? Stem tip. Here the root tip, you call it as coleorhiza. Then, right, here you can see that there is epicotyl above the level of scutellum, and the epicotyl has what? Has a short apex and few leaf primordia enclosed in a hollow foliar structure called coleoptile. Hope you have understood the structure of a dicot embryo and a monocot embryo. So, what is the structure of the embryo of monocot? Embryo of monocot possesses only one cotyledon, and that cotyledon is called as what? Scutellum, and is situated towards one end of the embryonal axis. And the embryonal axis has what? The radicle and the plumule. The radicle has what? A root cap, and is called as what? Is called as coleorhiza. Right, and there is epicotyl above the level of scutellum, and there is one more structure called coleoptile. What is coleoptile? The epicotyl has a short apex and a few leaf primordia enclosed in a foliar hollow leaf-like structure. What do you mean by foliar? Leaf-like structure called coleoptile. So, this is coleoptile. Hope you have understood the structure of a dicot embryo and a monocot embryo. So, in today's session 5, what did you all learn? We learned about the post-fertilization events, that is a very important event called the process of formation of an endosperm and the development of an embryo, followed by the structure of a seed, that is we studied about an albuminous seed and ex-albuminous seed. Then we studied about the structure of a dicot embryo and a monocot embryo. In my coming 2 more sessions, what are we going to deal with? In my coming sessions, in the next sessions, I will be dealing with the seed and its type. As I said, we will be learning about the albuminous seed and the ex-albuminous seed with suitable examples in detail, and what are the benefits of the seed and how do we protect them, all that we will be studying. Then as I said, the ovule develops into what? Seed, and the ovary will develop into what? Fruit. So in the coming session, 2 more sessions of this chapter called sexual reproduction in flowering plants, you will be studying about a fruit. Fruit is nothing but, as I said, the ovary develops into a fruit, and we will be studying about the types of fruits and its types, followed by apomixis and polyembryony in my coming 2 more sessions. Hope you have understood the concepts what I have taught you all in today's session 5 of sexual reproduction in flowering plants. So till we meet in the next session, till then, thank you.